Unit 8, page 5, mutations. How do mutations affect organisms? Do now. Define mutations. Pause the video here and write your response on your paper. Press play when you're ready to begin. Mutations are changes in DNA that affect genetic information. Some types of mutations. A point mutation is a change in a single base pair in DNA. A change in a single nitrogenous base can change the entire structure of a protein. You'll see in this figure here, we have a normal mRNA molecule that has been transcribed by the DNA and now translated into amino acids which make a protein. This entire structure of the protein is going to change just by changing one of these bases, changes the amino acid, and therefore changes the protein. Point mutations are changes in one or few nucleotides. For example, obviously these are not the ATGCs, these are actual sentences to show you how the changes would occur. So for substitution, you have the fat cat ate the rat. Now in your head you should be picturing a big fat cat that's eating a rat. Now if we change just one letter, you'll see how the whole meaning changes. The fat hat ate the rat. Now you're picturing a hat trying to eat a rat. Very different picture painted. Insertion. We have the fat cat ate the rat. Then we have the fat cat XLW ate the rat. So we can see how an insertion is going to take away from that meaning as well. And then a deletion. We have the fat cat ate the rat as opposed to the fat ate the rat. Gene mutations can also occur in what we call frame shift mutations. This is going to shift the reading frame of the genetic message so the protein may not be able to perform its function. Do you remember how many mRNA molecules make up a codon? That's right, it's 3. So if we shift that over and we're reading a different set of 3, again, we're going to change the whole meaning. So we can see again that same sentence, and if we insert to cause a frame shift, the fat, and then you'll see it's just letters because we just inserted an H, so we inserted one letter and we shifted the frame over so that these codons now don't make sense. Same thing happens with the deletion where we can shift them over to the left and we can see how it changes again the whole meaning of the sentence. So frame shift mutations are a mutation in which a single base is either added or deleted from the DNA is called a frame shift mutation because it shifts the entire reading of the codons by one base, which is then going to change all of those amino acids subsequently. This mutation would cause nearly every amino acid in the protein after the deletion or insertion to be changed. So obviously frame shift mutations are a huge concern as opposed to just a point mutation. Both can be bad, but frame shift obviously is going to have more of a profound effect. Chromosomal mutations, this is when an entire chromosome is going to have a problem. So here we can have the chromosome can break and lead to a rearrangement that produces genetic disorder or even cancer. So there are 4 different types of rearrangements. We can have deletion of one gene or several genes or just a piece. We can have duplication where there's a part that's repeated. We could have an inversion where the genes are flipped. So you can see here how they're flipped around. Or even translocation, where they end up on a different chromosome. Significance of mutations. Most are neutral, things like eye color, blue eyes, or light color hair are actually genetic mutations that are perfectly benign. Birthmarks are benign, they're not harmful. However, there are some genetic mutations that are harmful. Sickle cell anemia is a genetic mutation of just one amino acid being changed and obviously has a lot of severe consequences. Down syndrome is where there's an entire extra chromosome. It can cause a lot of different problems that we're going to get into briefly. However, some beneficial. Sickle cell anemia does give a person immunity to malaria. And if somebody does have a CD4 mutation, they're actually immune to HIV, as we had learned back in Unit 5. So with Down syndrome, chromosome number 21. So remember, you have 23 pairs of chromosomes. So think about chromosome number 21 is not properly separating during meiosis. So instead of that sperm or egg having 23 chromosomes, one of them will actually have 24. So therefore, when the egg and sperm join, instead of having 46, you'll have 47. As a result, children with Down syndrome are going to develop slower. They may have heart, stomach illnesses, and they vary greatly with their degree of intelligence, but it absolutely can have cognitive effects. So what are some of the causes of mutation? Well, there are two ways in which DNA can become mutated. Mutations can be inherited, meaning an exact copy of the mutation from the parent to the child, or mutations can be acquired, meaning that they happen over the lifetime. This happens with environmental damage or mistakes when DNA is copied. Any agent that causes DNA to change, we call a mutagen. So in summary, we have a DNA sequence here, TAC, CC, ACT. Find the RNA and amino acid sequence for the DNA strand above. Use the codon chart here to help you find the amino acid.